I invite you, I welcome you once more to my video channel. As usual, I like to establish that relationship, that symbiotic relationship between you, the public, and the medical laboratory scientists for a better health, a sound health. We all know that knowledge is power and health is wealth. When we combine the knowledge I shall impart with the wealth you shall bring from the better health, then you have power and wealth. Today's topic shall dwell more on semen analysis, knowledge review, and the laboratory diagnosis. When we talk of semen analysis, there is a need for us to understand what we are talking about. Semen has to do with the sperm we produce. And ordinarily, when you look at sperm production, you, after probably masturbation or sexual intercourse, you want to believe that everything is well. But that semen that you have produced may contain nothing that can cause that necessary pregnancy, that reproduction that you want. You probably are aspiring to become a father or a mother tomorrow. And the only way it can happen is when the semen contains the right proportion of things. The sperm cells, the spermatozoa are in large quantity, because so many of these sperm cells will be destroyed on their way to fertilize the ovum, and the environment around the uterus, around the journey towards the ovum, where it is located outside the fallopian tube. It's a very dangerous one because it kills so many sperm cells. And if one is by chance unlucky to also find antibodies against one's sperm cells, then that will not happen. But we are now talking of a situation where the sperm cells themselves are defective, or there is none. You don't even have one sperm cell. In such a situation, it is called azoospermia. The person does not have sperm produced. Even though the quantity of the semen produced is large, the patient or the donor of this semen has abstained as necessary for collection of the semen. We always advise that for collection, patients should abstain from sex or anything, even masturbation, for 7 days before producing this semen. So that quantity produced will let us know how the quantity normally produced by these patients, and then the quantity of spermatozoa in that quantity is reflective of what can fertilize the ovum. Those who can embark on this journey to take the golden fleece, that is, fertilize the ovum. It's a battle. Life is like a survival of the fittest life. It is the fittest that will eventually fertilize the ovum. So when there are no sperm cells in that semen that was produced, it's just nothing. There's no journey, there's no battle of survival of the fittest. It is from ab initio zero situation. Infertility confirmed, diagnosed as infertility, which might be caused by other problems, like the varicose vein blocking the production, which are further tests that will be done. It is not an immediate test. The semen analysis is just to know what is the situation of your semen, what you produce, what is contained inside it. So that is when there is no sperm cell, it is called azoospermia. There is oligospermia. Oligospermia is when you have not enough expected to be normally present in the donor, that is in the man. It is a very common statement you hear from some women that say, oh, my husband has low sperm count, or people say it's low sperm count. That is where the situation of the semen is, the proportion of semen in spermatozoa in the semen is below the normal level. So when you count them, they are probably in the region of 4, 5, 10 million, whereas you are expecting up to 80 to 150, 170 million sperm cells by males. So that really tells you what the chances of that man to really impregnate a woman is. So you can easily diagnose that the person is suffering from Oligoscamia, what you would call low sperm count. We also have situations where the sperm cells themselves, they have problem. There is a problem of where the shape of the sperm cells are not normal. The normal shape of the sperm cell is oval. This oval shape and the tail you see regularly. But where the proportion of abnormality of sperm cell is so high, then you are sure of a situation that is called teratospermia, that is the proportion of abnormal sperm cells in the region of 32% and above, and such situations you can imagine competing to fertilize the ovum with effective spermatozoa. The chances are you might not be able to impregnate the woman. So the cause of that situation will also be treated. But as far as my knowledge imparting today is concerned, it is limited to semen analysis. The problem of, the content of your semen. The other aspects might come in future presentations. So you can also have a situation where the sperm cells, though they are normal shape, but they are not moving progressively. They are not moving progressively as if they are on a mission, moving straight on a straight line, just going. And if they have to circle, they create that wide circular movement. They are not sluggish, because some are sluggish. That is another situation that is called spermia, where the sperm cells are sluggish and not making progressive movement. We have two types of movements, the progressive movement and non-progressive movement. The progressive movement, the chances are higher than the non-progressive movement, that probably you are telling somebody to run 100-meter race and is running backward. That will never win the race. It is very clear. But when you are doing a progressive movement, you are moving towards the touch line, moving towards the ovum. And that is very important for us to know that patients could suffer from problems like that. These are many more I shall expose. But as we continue along, we need to understand the need for us to like this program. Like, subscribe, so that quality production might come from you. We shall go now into the proper laboratory diagnosis, what the appearance and every other thing about semen analysis is about. But this preamble that I've given you is just to let you into what could defer or create that male infertility for most men. The women aspect is also there, but this semen analysis has to do with men, because it's only men that produce semen. Women don't produce semen. What you are going to understand about the female aspect is more on infection and hormonal imbalance and other things that could affect the sperm when it is produced in the woman. Because I spoke earlier about antibodies produced by the human. There are other acid, hyaluronic acid, that is produced by a woman that makes the environment almost hostile to the sperm cell. But that is not for real the topic right now. The topic right now is, let us move into the laboratory diagnosis of semen analysis, the knowledge and the review, so that we acquire more knowledge, have better health, have more power. Thank you for listening. Semen analysis and knowledge review. The lab diagnosis is a very important aspect of this whole procedure. The first aspect is, how do you collect a sample? You collect the sample after abstinence for 3 to 7 days. Collection into sterile bottle, clean, dry, and leak-proof, is very necessary. And you label the specimen bottle with date and time of collection. Within 1 hour, this sample must get to the laboratory and it must be examined within the serious period. The microscopic examination is just what you make sure that rhymes, that the labeling of the bottle tallies with the information on the request form. It must contain information of where the sample is coming from. You record the time examined, the color of the specimen, the viscosity, the volume, and the pH. The pH is usually 7.2. The microscopic examination is when you use the microscope, and usually we use 10 times 10 and times 40 objective for more vivid examination. The dilution of the sample is 1 in 20 in sodium bicarbonate formalin, and that would allow the count to be very easy. That is, you get up to 20, anything greater than 20 million is showing fertility. You also stain the blue, methylene blue will always stain the nuclei of the spermatozoa, and the semen is also tested for presence of fructose, which if blocked shows that there is a problem with the seminal vesicle. The head of the sperm cell, as we see from this picture, is that aspect that contains the nucleus, and it's always blue when stained. The situation of astenospermia that I spoken about has to do with the type of motility. When it's grade A, then that is not astenospermia. It moves very fast to the point of where you get it. The phase contrast advantage is shown here. The phase contrast is the middle one. You can see the sperm cell appearing vividly. It is better than the dark contrast or the light microscope. WHO slide examination is what we have now seen. We see the sperm cells all around the swim.